This is OwlUp, where learning happens in minutes, not hours. For a deeper journey, explore all our courses at owlup.co. Now, let's dive in. Welcome to Chapter 1 of our series, Mastering Lab Techniques Safely. In this episode, titled Introduction to Lab Safety and Equipment, we'll explore the common hazards you might encounter in the lab and how to navigate them safely. So, let's dive in. A burner hisses on the crowded lab bench. A new student leans in, barefaced, reaching across the open flame toward an unlabeled bottle, sneaker heel grazing a coiled cord on the floor. In this frozen second, here's the real question. What stops this from becoming a headline? That frozen second is shaped long before anyone turns on a gas tap. Modern lab safety starts upstream, in how experiments are designed, how equipment is chosen, and how the workspace is set up before day one. A clean bench with clearly labeled reagents, grounded outlets, and unobstructed exits prevents the near miss from ever forming. In a teaching lab, that means assigning roles. One student focuses on pipetting, another on recording data, a third on monitoring shared equipment, so no one is stretching across hazards. In a research lab, it means formal risk assessments, written SOPs, and pre-use checks. Is the fume hood's airflow in range? Are the correct gloves stocked? Are incompatible wastes separated? When these habits become routine, the hissing burner is just background, not a looming threat. In real labs, safety decisions are rarely dramatic. They're dozens of small choices made in seconds. A chemist pauses before opening a new solvent and checks the SDS for flashpoint and inhalation risks. A microbiology student notices moisture inside an incubator and wipes it down with disinfectant before loading cultures. A physics tech testing lasers posts a Class 3B in use sign and verifies interlocks before aligning optics. These actions rely on knowing which hazards exist, which PPE matters most for that task, and which piece of equipment actually controls the risk at its source. A safe lab day really hinges on 3 questions you ask before you touch anything. 1. What can hurt me here? 2. How will it try to hurt me? 3. What stands between me and that hazard? Those answers change with the task. Start with inherent hazards. Chemicals can burn, poison, corrode, explode, or quietly damage organs over time. A bottle of concentrated nitric acid and a bottle of diethyl ether may look equally harmless on the shelf, but one wants to eat through tissue, the other wants to evaporate and ignite. In a chemical synthesis lab, that means checking which reagents are flammable, which release toxic vapors, and which react violently with water or air. In contrast, a cell culture lab centers on living hazards, bacteria, mammalian cells, viral vectors. Here the concern shifts to routes of infection, splashes to eyes, aerosols from vortexing, accidental needle sticks. In a laser optics room, eyes and skin are targets, and reflections from shiny tools can be just as dangerous as the direct beam. Next, connect hazards to controls. For volatile solvents, the primary control is a functioning fume hood. PPE only backs it up. For infectious aerosols, a certified biosafety cabinet with correct airflow is non-negotiable. For cryogens like liquid nitrogen, insulated gloves, a face shield, and ventilation prevent frostbite and asphyxiation. In each case, PPE is chosen to match the hazard. Splash goggles for corrosives, impact-rated eyewear for flying glass, cut-resistant gloves when handling broken glass or blades. Equipment choice then shapes how safely you can work. A chemistry student weighing cyanide salts uses a balance inside an enclosure, not on an open bench. A microbiologist streaking plates keeps the Bunsen burner away from alcohol waste and uses a heat-resistant pad under it. A material scientist cutting metal samples chooses a saw with a guard and a local exhaust line, not a handheld tool pointed toward the room. The pattern is simple but powerful. For every task, identify the specific hazard, choose the right engineered control, and only then add task-appropriate PPE as your last, not first, line of defense. At UCLA in 2008, a chemist working with pyrophoric T-butylithium transferred it in a regular syringe, outside a fume hood, wearing only a lab coat made of synthetic fabric. When the reagent ignited, the coat melted into the skin. That single event reshaped university safety programs worldwide. Flame-resistant lab coats became standard, training for air-sensitive transfers expanded, and written approval was required for high-risk reagents. In a teaching lab, a more routine example, a student caps a tube of volatile solvent tightly and leaves it on a warm hot plate. The pressure builds, the cap pops, and solvent sprays. Because the lab adopted splash goggles and kept all heating zones clearly marked on bench diagrams, no one is injured, and the incident becomes a case study in the next safety briefing. In a BSL-2 microbiology course, instructors now integrate near-miss reporting. Students anonymously log events like unnoticed glove tears. Over a semester, patterns reveal which procedures need redesign. Building on those examples, here's the deeper shift. Lab safety isn't about avoiding trouble with the instructor, it's a professional habit and a quiet form of respect. Every time you wear goggles or use the hood correctly, you're protecting lab partners, custodial staff, and the next person who walks into that room. Have you noticed how the calm, prepared people in a lab get trusted faster? In your next class, internship, or research role, the ones who can follow protocols confidently are the ones who get more independence and earlier. There's often a silent resistance. The PPE is annoying, or I'll remember the emergency steps if something happens. But here's the key idea. The small discomfort of a lab coat or taking an extra half-minute to find the spill kit is nothing compared with the hours or days lost if an avoidable accident sends you or a colleague to occupational health. So, as you move into your next lab, try this mindset. Each checklist you follow, each label you double-check, is part of your reputation. You're showing that you take other people's safety as seriously as your own, and that's exactly the kind of scientist, engineer, or clinician people want beside them when something unexpected happens. With that foundation of safety as professional respect, here's the framework to carry forward. Pause, protect, proceed. Pause to scan for hazards. Protect with the right PPE and protocols. Proceed only when your equipment and setup match the risk. Picture that student again. Now they stop, gear up, reroute cords, and adjust their setup before reaching in. That tiny reset turns a near miss into a non-event. Your challenge this week: before your next lab, actually walk the room. Find the eye wash, shower, and fire tools. Confirm what you're expected to wear, head to toe. Then, on purpose, spot one hazard you'd overlooked, maybe a blocked exit or a crowded bench, and decide how you'll handle it next time. This was OwlUp, your daily step forward. To keep learning with structured courses and more insights, go to owlup.co. Until tomorrow!